Hi dear students. So in this video lecture, we will be talking about the organization of cells and their subcellular organelles. So guys, as we all know, what is cell? Cell is a functional and the structural unit of living beings, right? Like humans have almost 37 trillion cells. But if we broadly classify the cells, we have two types of cells, prokaryotic and eukaryotic cells. So what is the basic difference between these two? As we all know guys, size of the prokaryotic cell will be smaller, but eukaryotic they will be variable, right? Then number 2, nuclear membrane is absent in the prokaryotes but present in the eukaryotes. Number 3, guys, there are organelles, membrane bound organelles. These are absent in prokaryotes, like they don't have lysosomes or peroxisomes or mitochondria. Fine, but eukaryotes they have a membrane bound organelles, right? If we talk about the composition of plasma membrane, cholesterol or sterols are only found in eukaryotic cell membrane. If we talk about the type of ribosomes, then as we all know that prokaryotes they have a 70S and the eukaryotes they have a 80S type of ribosomes. Fine. Then the location of electron transport chain enzymes is in the plasma membrane of prokaryotes but in the inner mitochondrial membrane of the eukaryotes, right? Cell division in the eukaryotes is very complex, right? Like mitosis are involved here. Fine. But binary fission can be the simple mechanism in the prokaryotes. Correct guys? The extra chromosomal DNA in the prokaryotes, that is plasmid, but in the eukaryotes it is, what do you call it? In the eukaryotes we have mitochondria, and mitochondria they have a separate DNA, right, apart from the nuclear DNA. And guys, if we talk about the nuclear DNA again, histone proteins are only found in the eukaryotes but not in the prokaryotes. Correct? Now guys, cells and cell organelles, right? Basically the cell organelles can be separated by a process that is called as centrifugation. And this process is also called as subcellular fractionation. Okay, so we'll use a high-speed centrifuge here, and in the centrifuge guys, we all know it is based on the density. So those who are the denser organelles like nucleus and mitochondria, they will settle down, and those which are having lesser density like lysosomes, they will appear on the top. Fine. Now guys, during the centrifugation, right, when we study the cells, we can identify the cells based on their marker enzymes, right? So subcellular organelles, they have a particular enzyme and we can identify them by that enzyme. See guys, what is the purpose of having, first of all, these subcellular organelles like lysosomes, peroxisomes, mitochondria, etc. See, the point is that the various chemical entities can be concentrated at a particular place. Let's say you want to do a reaction in mitochondria, so that particular chemical reactants will be accumulated in the mitochondria. So we are providing a environment which is enclosed, right, which has a higher number of chemical reactants. Okay, so we can start the reaction to produce the product. Okay, instead of doing the same reaction in complete open cell. So these subcellular organelles can be the site where the concentration can be built, right, for these chemical reactants to get the chemical reaction, right, and a smaller concentration we can get the products, desired products which are required for function of the cell, right? Talking about the marker enzymes, guys, the marker enzymes are the enzymes which are specifically found in a particular organelle. For example, how will you identify the plasma membrane? So marker enzyme is sodium potassium ATPase, right? And it's a pump, sodium potassium pump. Then the second is like mitochondria. So ATP synthase, the best function of the mitochondria can be described as ATP synthesizing machinery, right? Then number 3, we have lysosomes, and like acid phosphatase is one of the enzyme there. In the peroxisomes, There's a name of enzyme which is called as marker is catalase because it breaks the hydrogen peroxide. In the endoplasmic reticulum, guys, the name of enzyme is called as glucose 6 phosphatase. We'll discuss this enzyme many times during the lectures. This enzyme will be there in the glycogen metabolism, in the gluconeogenesis also, right? So we'll see that enzyme again. Then, guys, we have the Golgi apparatus, and in the Golgi apparatus, we have galactosyl transferase. Okay, because Golgi apparatus is mainly involved in the process of adding sugar or making the glycoproteins. Adding sugar to the proteins. So that's a kind of modification of the proteins once they are synthesized. Okay, so guys, these are the marker enzymes that you should remember always because they're very important in your viva or maybe in your theory as a short question, right? One question maybe of just 2 marks. Now moving to organelles one by one. So guys, the first and the most important organelle that we have is the nucleus. Nucleus, right? What is the function of nucleus, guys? The function of the nucleus is the storage of the DNA or the genetic information. The second function is like, let's say, the transcription, the making of the mRNA, which will be delivered to the cytoplasm for protein synthesis, right? Nucleus has 2 membranes, the inner nuclear membrane and the outer nuclear membrane. The outer nuclear membrane is, guys, continuous with the membrane of endoplasmic reticulum, right? As you can see. Fine. And guys, only the eukaryotic DNA, they have the histone proteins. Fine. Histone proteins are present in large quantities. They are the basic proteins and positively charged, which can easily bind with the negatively charged DNA. Right? But remember, guys, sperm has no histone. Human sperm, if we talk, it has no histone proteins, but some other proteins will be present, which are basic and positively charged. Right? Now, moving to our next point, next organelle. So guys, next we have the endoplasmic reticulum. Now guys, endoplasmic reticulum can be subdivided into 2 parts, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Now in the rough endoplasmic reticulum, why they're called rough? Because they are studded with ribosomes. Okay, the protein synthesizing machinery. So they have the ribosomes, so that is why, basically their function is to make the proteins. And guys, these proteins are specifically made, right, that they will be given to lysosomes or they will be secreted out of the cell. You got my point? Secretory proteins are made by the ribosomes which were present on rough endoplasmic reticulum. Okay, cytosolic ribosomes are also called as free ribosomes. Fine. So they are making proteins like, which will be transferred to, let's say, mitochondria, nucleus, peroxisome, etc. Fine. So that is your cytosolic free ribosomes. Fine. So we have also discussed the ribosomes right now. Fine. So guys, that is rough endoplasmic reticulum. Then guys, we have the smooth endoplasmic reticulum. Smooth endoplasmic reticulum has a function of lipid synthesis, right, and steroid synthesis. Fine, guys. And also they have a very important function that is called as detoxification or the xenobiotic metabolism. You know why? Because they have the CYP protein, cytochrome P450 monooxygenase protein, or the enzyme is found there, right, which helps in detoxification of the foreign substances like drugs, like pollutants, like preservatives in the food, etc. So that is one of the function of smooth endoplasmic reticulum. Also, guys, in the muscles, they are the site of storage of calcium, so which is required for muscle contraction. So lipid synthesis, detoxification, storing the calcium. So these are some of the function of the smooth endoplasmic reticulum. Now guys, moving to the next point, that is Golgi apparatus. Just remember guys, Golgi apparatus, these are just like stacks of the membranes. Fine. And these Golgi apparatus, they are very important for secretion of the protein out of the cell, right? So the secretory pathway of the endoplasmic reticulum, which has started, is ending with the Golgi apparatus making the vesicles, right? And these vesicles, they will be delivered out of the cell. So that is secretory pathway is going like this. But the second very important function of these Golgi apparatus is what? Is a post-translational modification. Guys, these are the most important sites for adding sugar to the proteins. Like the O-linked glycoproteins are specifically made only in Golgi apparatus. And the N-linked glycoproteins are made in both endoplasmic reticulum and the Golgi apparatus. So what we did here is protein was made by ribosomes only, but we just added the sugar. That is glycosylation we did. So O-linked glycosylation. And that's why what was the name of enzyme? Galactosyl transferase or glucosyl transferase, the marker enzymes. They are adding sugar to the proteins. The O and N means simply, guys, sugar will be added to the OH group of the amino acid or the protein. That is O-linked. N-linked means the sugar will be added to the nitrogen of the proteins, like asparagine amino acid, for example, is a site of N-linked glycosylation. So that is all done in the Golgi apparatus. That will be discussed somewhere else in other chapters, right? Okay. Now moving to the next point. Okay, so next we have lysosomes. Lysosomes are single membrane structures, right? In their membrane they have proton pumps, right? H plus protons. And guys, this proton pump is simply maintaining the pH of the lysosomes at around 5. And that's why the most of the enzymes which are found in lysosomes are called as acid hydrolases. So which class of enzymes are present in lysosomes? Class 3, that is hydrolase. Okay, hydrolase is the name of the enzyme that is found in these organelles. What is the function of lysosomes? Guys, lysosomes are simply called as the organelles which digest, right, the materials which is maybe cellular or extracellular. Okay, so when the cell, let's say when the cell takes something from outside by the phagocytosis or the pinocytosis or the endocytosis, whatever, so the vesicle which is entering into the cell will fuse with lysosome, and that's how the secondary lysosomes are formed. So vesicle carrying some material, from the cell or out of the cell, right, will fuse with the lysosome. Now lysosome has various enzymes which are hydrolases, like nucleases, DNAs, RNAs, like they have the lipases, like sphingomyelinase, then they have the proteases, like cathepsins and collagenase, then they have carbohydrate digesting, enzymes, right, like hexosaminidase, for example, if I say. Okay, so there are various enzymes are found in the lysosomes which are degrading everything possible, fats, lipids, proteins, or carbohydrates, or even the nuclear material, right? So lysosomes are very, very important organelles in this term. But guys, they can be dangerous also. If these enzymes are released without any reason, right, or if the lysosome membrane is damaged, then they can cause the inflammation or damage to the surrounding tissue. This is what happens in, let's say, the gout, a type of joint disease. In the gout, which is a type of a disease, right, which is a joint disease where the uric acid crystals are taken in the lysosomes, and they may rupture the lysosomes, causing the potent hydrolase enzymes to come out in the surrounding and causing damage, inflammation, pain, etc. Fine. And guys, even during post-mortem, right, when the person is dead, right, so in the dead body, the post-mortem changes happen, and one of the earliest post-mortem change is actually what? That lysosomal membranes are damaged, right? They rupture, and these lysosomal enzymes, they are out. Okay, and that's what you call as autolysis, right? Okay, so that is post-mortem autolysis. That means lysis of the body, decomposition of body has started, simply speaking. Clear, guys? Fine. Now let's move to our next point. So let's say we have now peroxisomes. Okay, guys, peroxisomes are very important organelles. Why? Because they have catalase enzyme, and catalase enzyme destroys the very strong oxidant, that is hydrogen peroxide, H2O2. So H2O2 is degraded by this catalase enzyme, right, preventing any damage, further damage by this H2O2, hydrogen peroxide. That's an oxidant basically, right? Okay, now guys, peroxisome is also a very important site of certain pathways. like bile acid synthesis, for example. Okay. Then the second is the beta oxidation of very long chain fatty acids, right? Fatty acids which are more than 22 carbons, that is very long chain fatty acid. We'll discuss this everything. Right now we have to just do the basics, that what are the pathways taking in peroxisomes. Alpha oxidation of fatty acid like phytanic acid, which is found in the milk. Okay, phytanic acid. It is different from phytic acid. Phytic acid is different. That's not phytanic acid. Phytic acid is something which is preventing the absorption of the minerals because it is found in certain food items. So that is phytates. That's different. It's phytanic acid. Okay. So alpha oxidation of phytanic acid, beta oxidation of very long chain fatty acid, bile acid synthesis, and plasmalogen synthesis. Guys, these are type of ether lipids. Plasmalogens, like which are found in large quantities in the heart. Plasmalogens. Correct, guys? So these are the basic pathways which are taking place in peroxisomes. And guys, if a baby is born without peroxisomes, without functional peroxisomes, if I say, then that disease is called as Zellweger syndrome. Okay, Zell, Zellweger syndrome. So that is a disease which is affecting the cerebrohepatorenal tissues. That's why we also call it cerebro means brain, renal means kidney, and hepato means liver. Cerebrohepatorenal syndrome, you call this. Okay, guys. So that is your Zellweger syndrome. That name you should know. Various other disorders we will discuss in other sections of lipids, right? Okay. So that is all about peroxisomes. Now, what is the next organelle, guys? Very important, mitochondria, right? The mighty mitochondria. Guys, the mitochondria is having again two membranes, outer and inner. Outer membrane is permeable to a lot of things, right? Easily, but inner membrane is selectively permeable. And it has some transporters also. The space between the two membranes is called as intermembrane space, which has its own, utility that we'll discuss later in details in certain other pathways, right? Now, what was the name of the enzyme, which was a marker enzyme? ATP synthase, right? Which is the main enzyme of mitochondria making ATP. So, guys, the center portion of the mitochondria is called as matrix, and the inner membrane is folded, right, increasing its surface area, right, and these are called as cristae. So when it is folded, it's called as cristae. So what are the pathways which takes place in the inner membrane of mitochondria? Electron transport chain, right, guys? Electron transport chain. And what are the pathways which takes place in the matrix of mitochondria? In the matrix of mitochondria, guys, a lot of pathways are taking place. Number one, Krebs cycle, the most important pathway, Krebs cycle, right? Then we have beta oxidation of fatty acids. Okay. Then partially, guys, also heme synthesis, urea cycle, and gluconeogenesis. They are also taking place in the partially in the mitochondria. When I say partially, guys, I mean to say that these are the pathways also taking place in other cell organelles, right? Okay, like for example, urea cycle starts in the cytoplasm. Fine. Urea cycle has also cytoplasmic reactions. Not doesn't start in cytoplasm, but it has cytoplasmic reactions also. Similarly, gluconeogenesis and similarly the heme synthesis. Fine. So these are all the pathways which are taking place in multiple cell organelles. We can remember them like as HUG. Hug, right? HUG, and hug simply means heme synthesis, urea cycle, and gluconeogenesis. Why I said hug? Hug simply means the two part of the cells are involved, right? Hugging each other. Cytoplasm, mitochondria. Just to remember. Fine, guys. Okay. So that is one of the important thing about mitochondria. Guys, one more very important thing about mitochondria is that they have their own DNA, which is double strand circular, similar to bacteria's. And the theory of mitochondriogenesis is also says that it's the bacteria and the other cell symbiotic relationship which resulted in the mitochondria in human cells, right? So this DNA, guys, it is able to express itself and can make around 13 proteins of electron transport chain. Right? Out of the 67 proteins of the ETC, the 13 proteins can be made by mitochondria itself. So it's a very, unique DNA that we have, which is similar to the bacterial DNA because it is circular and lying freely in the mitochondrial matrix. Right? And guys, because of the presence of ETC very nearby, there are a lot of electrons are released from ETC, which can cause a lot of mutations in this mitochondrial DNA. Fine. And guys, each cell has almost 1000 to 2000 mitochondria. And those cells which need more energy, like heart, for example, or the muscles, they have more number of mitochondria. Fine, guys. So that is all about our mitochondria. There is just one more term that I want to highlight here. There are two terms which are called as microsomes and microbodies in our cells. What are they? See, microbodies is simply the other name of peroxisomes, right? But what are microsomes? Guys, microsomes are nothing but disintegrated parts of endoplasmic reticulum during subcellular fractionation, during centrifuge, when the endoplasmic reticulum is disintegrated. So small bodies are formed and these are called as microsomes. And that's why we see, sometimes we see the enzymes are like this, microsomal cytochrome P450 enzyme. So that is microsomal, simply means the endoplasmic reticulum. Right? So microbodies are peroxisome. Fine, guys? Okay, so that's all about the basics of subcellular organelles. Right? I'll see you in the next video.